In this class, I am explain about microscope. Microscopy is a branch of science that deals with the study of construction, working principles, and use of microscopes. The microscope was invented by two Dutch spectacles makers, Hans and Zacharias Janssen, around 1590. They found that a combination of two hand lenses could greatly enlarge the image. Its combination of lenses gave magnification of the order of 50 to 100x. This was the beginning of the compound microscope. Robert Hooke in the 1660s developed a compound microscope with which he described the microscopic structure of cork cell in his Micrographia. He probably obtained a magnification of 200x, which is evident from his pictures. A contemporary of Hooke, who used his own simple microscope for describing microorganisms, was the Dutch philosopher Anton van Leeuwenhoek. Leeuwenhoek's microscope consists of a single convex, rather spherical lens mounted on a stand of brass or silver. The lenses were ground by him. The early microscopes were light microscope because they used light, either sunlight or light from an artificial source for observation. Today we have mainly two categories of microscopes: those in which light is used for observation, light microscopes, and those in which a beam of electrons is used for inventioning the objects. Light microscopes may be divided in the following categories: bright field microscopy, dark field microscopy, fluorescence microscopy, phase contrast microscopy. Electron microscopes can be divided into four categories: transmission electron microscopy, scanning electron microscopy, scanning tunnel electron microscopy, and immunoelectron microscopy. Other types of microscopy are X-ray microscopy, where X-ray are used, and autocasting microscopy, where sound waves are used. First one, light microscopy, bright field microscopy or compound microscopy. This is the most widely used type of microscopy. Here the microscopic field is brightly lighted and the microorganism mounted on a glass slide appear dark because they absorb light, resulting in greater contrast and color differentiation. Light microscopy generally enlarge up to 1000x to 2000x, and greater magnification than this leads to loss of clarity. The compound microscope of today usually consists of a strong metal stand with a broad base. The base contains a light source, either an electric lamp or a mirror. Above the light source is a system of iris diagrams which regulate the passage of light and the eliminate undesirable peripheral rays from the light source. Attached to the stand above the iris diagram is an adjustable condenser which concentrates the light rays on the objects. Above the condenser is the working platform or stage of the microscope with an opening at the center to admit light from the condenser. Glass slides containing Bacterial smear or mounts are to be centered on this platform. At the lower end of the vertical tube are the objective lenses. These are usually 10x, 45x, 100x. The 100x objective is the oil immersion lens to be used for the highest magnification, with a drop of oil between the lens and the material on the slide. The oil used is purified cedar wood oil of the particular consistency. The objective lenses are fixed to a revolving turret or nose piece, which may be rotated into position to bring the right objectives to face the material. The objective produces a real image of the specimen within the vertical tube. At the top of the vertical tube is the ocular lens or the eyepiece. This lens further enlarges the real image into a much larger virtual image. The structural component of compound microscope. The three basic structural components of a compound microscope are head, base and arm. Head or body, it is optical part in the upper part of the microscope. Base of the microscope support the microscope and the illuminator. Arm connects to the base and support the microscope head. It is also used to carry the microscope. Optical component. Eyepiece or ocular is what you look through at the top of the microscope. Typically standard eyepiece have magnifying power of 10x. And eyepiece or ocular. Eyepiece or ocular is what you look through at the top of the microscope. Typically standard eyepiece have magnifying power of 10x. Optional eyepiece of varying powers are available, typically from 5x to 30x. Next one, eyepiece tube. It holds the eyepiece in place above the objective lens. Binocular microscope heads typically incorporated a diopter adjustment ring that allows for possible inconsistency of our eyesight in one or both eyes. Next, objective lenses are the primary objective lenses are the primary optical lens on a microscope. They range from 4x, 100x and nose piece the objects are exposed and are mounted on the rotating turret so that different objectives can be conveniently selected. And Coras and fine focus knobs are used to focus the microscopes. And a stage is where the specimen to be viewed is placed. And stage clip are used when there is no mechanical stage. And aperture is the hole in the stage through which the base light reaches the stage. And illuminator is the light source for the microscope, typically located in the base of the microscope. Mostly light microscopes use low voltage halogen bulbs with continuous variable lighting control located within the base. Condenser is used to collect and focus the light from the illuminator on the specimen. And iris diagram, it controls the amount of light reaching the specimen. It is located above the condenser and below the stage. Most high quality microscopes include an Ebb condenser with an iris diagram. Next one, condenser focus knob moves the condenser up or down to control the lightning focus on the specimen. And working construction of compound microscope. Resolving power. The resolving power of a microscope is its ability to distinguish two adjacent points as separate. For example, 2 points or lines with less than 200 nanometer space between them are not distinguishable as separate lines or points to the unaided eyes. Mere magnification without the ability to distinguish minute structural details is not beneficial in a microscope. The resolving power of the microscope depends on two factors: the wavelength of the light and the numerical aperture of the lens system. The wavelength of the visible light used in light microscopy is 400 to 750 nanometer. Resolution is inversely proportional to the wavelength. Numerical aperture. The measure of the aperture of the objective is an angle 0 subtended by the optical axis and the outermost rays covered by the objective. Angle 0 is the off aperture angle. The magnitude of this angle is expressed as a sine value. Sine value of this off aperture angle multiplied by a refractive index n of the medium filling the space between the objective lens and slide gives the numerical aperture. If this medium is oil, the refractive index is 1.56. If it just air, n value is 1. The numerical aperture for oil immersion system is 1.3. The numerical aperture for the dry objective is less than 1. For oil immersion objectives, the value ranges from 1.2 to 1.4. Thus, the resolving power of light microscope is limited by wavelength of the visible light and the numerical aperture. The diameter of the smallest object on the least distance between two objects which may be resolved by a lens system is the resolving power of the microscope and may be calculated as follows. For example, resolving power wavelength is 600 nanometer, 2 into numerical aperture 1.4, that is 0.21 mu meter. And magnification. Most light microscopes have three objectives: low power, high power and oil immersion. The primary magnification given by multiplying the magnification power of the objective, that is 10x, 45x, 100x, by the magnifying power of the eyepiece, that is 10x, 15x, 20x. A magnification higher than the resolving power of the microscope will give hazy images. Spherical and chromatic aberration. Inequalities of refraction by the peripheral portions of the objective's lens results in spherical aberrations resulting in fuzzy image. chromatic aberration. Color is due to the prism-like effect of the outmost portions of the lens. In modern microscopes, spherical aberration is overcome by the ingenious design and curvature of the lens and partly by the use of the iris diagram. Lens system corrected for chromatic aberration in the red and blue ranges are called achromatic lenses, and those corrected in red, blue and other ranges apochromatic lens. Uses of compound microscope. Used to view creatures in soil, water and others. Used to see anatomical structure of plant, animals and cells. Used in clinical diagnosis of disease caused by bacteria, protozoa and fungi, etc. Thank you.